Okay, hi everyone. So yeah, it's true. So I recently joined the Restore Institute in Toulouse, not a year ago, actually less than that, maybe 6 months ago. And it's a pleasure to be here and talk to you a little bit about zebrafish and how can we explore its regenerative abilities in the context of biomedicine. So this morning I changed medicine to biomedicine, in fact, because I think it is more accurate, because in fact we don't use zebrafish in clinical settings, as you can imagine, but we do use it to try to identify or to try to answer fundamental questions that then later on might help us in clinical settings. So, and this is, sorry, it's not working. And this is what I call physiological regeneration. So zebrafish is really able to regenerate multiple organs. And it's not clinical regeneration, but this is what we hope to achieve by studying these regenerative organisms. So I'll just give a brief introduction about the fundamentals of regeneration to make sure that everybody's on the same page. And I'll tackle different aspects. So don't worry, because I know everybody's from different backgrounds and I know everybody will not understand every single aspect of what I talk. But I think the idea is to provide a notion of what we can do with zebrafish in terms of regenerative biomedicine. So regeneration in the field, it's considered a full restoration of the tissue upon damage, loss, or even degeneration. So this means that the tissue is restored in terms of function. So it functions as the original tissue, and it has no permanent fibrosis or scarification. So this is very limited in humans. So we still do have some ability to regenerate some organs. For example, the skin, we do regenerate it to some extent. The liver is a regenerative organ in humans and mammals in general. And we also regenerate the blood. So we keep renewing our blood cells. Now, regeneration can take place in different levels of biological organization. So cells themselves, for example, axons, they can regenerate, for example, their dendrites. Then we can start going up in more complex organization levels. So we have simple tissues such as epidermis. As I told you, we are still able to regenerate part of the epidermis. Also the gut lining, we do regenerate to some extent because it suffers constant abrasion. And then we go to more complex settings. So we have internal organs like the heart, structures which are composed of many different tissues, and also whole body regeneration. This is what happens, for example, in planaria. So there are multiple outcomes post-injury in terms of regeneration. And here I'm showing you regeneration and repair, because these are in fact different things. So as I said, regeneration implies the full restoration of tissue with its shaping, with its patterning, with everything until it's fully functional again. With repair, that's not so much what happens. Usually there's scarification and to some extent a little bit of cell proliferation, but not full regeneration. And again, if we go deeper to the bad, let's say, spectrum of post-injury outcomes, if we have, for example, chronic inflammation, this is very, very bad, and it tends to lead to exacerbated fibrosis associated with chronic wounds. And this doesn't mean that inflammation, in fact, is not required for regeneration. We do need, or regenerative organisms do need, an acute self-limiting inflammatory wave. So this actually triggers regeneration. The problem comes when this inflammation becomes persistent or chronic. And this is where the regenerative potential decreases, while the inflammatory response increases. And it's very important, this in terms of aging, because with inflammation taking place as we get older, our inflammatory potential increases and regenerative potential decreases. So there are multiple types of regeneration in the animal kingdom. We have what we call the stem cell mediated regeneration. We have, this is what happens in the blood, as I mentioned before. We have morphallaxis, and if you think of hydrus, this is what happens. So for example, if you cut one of these organisms in half, what happens is that the two halves will reorganize, so these cells will reorganize into smaller organisms, and then these organisms will then grow again. So we have compensatory regeneration, the liver, for example. So if you cut a bit of the liver, it still regenerates. However, we know that the liver has two lobes, for example, and if you cut an entire lobe, you will not have a new lobe being formed. What you have is that the remaining tissue from the remaining lobe will enter, the cells from this tissue will re-enter cell cycle and will regrow, but in a single lobe. And this is what happens, for example, when people donate a little bit of the liver, and the recipient will grow a single lobe, but it's still functional. And then we have epimorphosis, and this is mostly what zebrafish does in most of the organs. And I'll go a little bit deeper on this. So epimorphosis implies that upon an injury, so we kill some cells, there's the formation of a blastema. So this is an intermediate or a mediator structure. So what happens here is that the cells around this injury, they will actually de-differentiate. So they'll lose their mature identity in order to be able to proliferate. So they'll de-differentiate, undergo endothelial to mesenchymal transitions, and then they will proliferate to fill in, this gap of tissue. They'll proliferate and then slowly re-differentiate again in the new tissues. Okay, so zebrafish does this in multiple organs, such as the heart. The nervous system is very important as well, as you can imagine, in terms of neuronal tissue disease and traumatic injuries, such as the spinal cord, for example. The lateral line, also very important, as it is a model for the loss of hearing cells, pancreas, kidney, liver, mandible, everything you can imagine, zebrafish will probably regenerate it. And also the caudal fin, which is an ex libris of regeneration in the field of regenerative biology. And you will understand why. So I will not talk about all of these. I'll focus on a couple of those, so mostly the heart and the caudal fin, but I hope it gives you an idea of the potential of using zebrafish to understand regeneration. So as I said, the regenerative potential declines in mammals as opposed to zebrafish, for example. We still have some regenerative ability in some organs, but this becomes a problem when we look at the heart and the bone, for example, which we hardly regenerated. But the zebrafish does regenerate multiple of these organs, and the fin, as I said, an ex libris, as you can see here, regenerates very fast. So in 2 weeks, you almost have a fully regenerated fin. So what we do is we amputate here, and the fin regrows. So why the fin? So the fin is actually histologically simple, as you can see here. So it's composed of mineralized bony rays here, as you can see. And if you go deeper in the tissue, you see that these are basically two hemirays together. And if you make a cross section there, you see that we have epidermis on the outside. We have the bony rays there in red, which are actually covered by osteoblasts, which are the bone forming cells. And in the middle, we have a mesenchyme and also blood vessels and pigment cells. So it's simple, but because it's simple, it allows us to study fundamental questions about regeneration, but it still holds complex patterning mechanisms, and it has shared developmental mechanisms with mammalian limbs. And of course, we can easily amputate it without killing the animal and then track the regenerative process. So what happens when we amputate this fin is that the cells below the amputation plane, the epidermal cells, they migrate, and this is very fast, and they form like a cover. So we call it a wound cap. And then what happens is that the remaining cells from the below tissue will undergo dedifferentiation, as I said, and then in the tip of each ray, they will fall an individual blastema. And then slowly, this blastema starts redifferentiating in the new tissues, and we have mature tissue on the proximal positions, and we keep a blastema at the distal points until the regeneration is fully complete. So this blastema, or this regenerating tissue, is highly organized. So here on the tip, we have the blastema, as I said, and then we have a gradient of differentiation. from distal to proximal positions. And here in blue are the bone arrays, and we have different markers, for example, for the bone cells. So RUNX2 are more immature, and then you have osteocalcin here in more mature positions. And this is what you see here, for example, with these reporter lines. So we have these at 3 days, for example, we hardly have any mature bone cells, but we have a lot of them immature. So you can track all these things, and because it's a very thin structure, we can actually image what happens in terms of cells. So as I said, the cells de-differentiate. So this here in green are the osteoblast cells. So these are mature cells, and you see they undergo endothelial to mesenchymal, epithelial to mesenchymal transition, and then they migrate here to this plastema, where they are de-differentiated, and then they slowly redifferentiate again. And I hope you can see on this video here that, yeah, so you have an osteoblast here from below the amputation plane, and it's de-differentiating and migrating to form the plastema. So not only we can use this system to understand the fundamental mechanisms of regeneration, but because it's the formation of new tissue in an adult organism, we can actually understand how these new tissues are formed. And one aspect of these bone arrays is that they do bifurcate. So one question that I had in the past was, how are these bone structures actually being shaped? So it provides us a nice tool to address a very simple question: how does a bone array bifurcate? So in the model back then was that we have bone apposition taking place after the injury. So the tissue is growing, bone is being formed, and then at some point it splits, right? And there's some signals doing that. And then it keeps growing. Okay, so what I found back then, what we found is that this is not necessarily the case. So indeed, there is a splitting here. So if you follow this same array over time, which is something that we can do with this system, you see that it splits there at 1 day, but 2 days later, it's actually there. So there's indeed a splitting mechanism, but there's also a stitching taking place there. So then where is the splitting point? Where is the branching point in the end? So we found out, okay, there's a stitching, so there must be an anti-stitching activity. So we looked at reporter lines that we had available back then that typically label what we call osteoclasts, and these are the cells that actually eat bone. And we see that they are there before injury, before splitting. But then once the branching point is defined, what we see is that there are these elongated forms here that we found and we called osteolytic tubules that line on the surface of bone. And if we stop their bone resorbing activity, because they do have bone resorbing activity, we see this with the staining there. And if we block this bone resorbing activity, what happens is that these rays do not bifurcate. So they never split. So with this, we found that there is, in fact, a stitching of these splitting bone arrays, and there is also the counteractivity of osteolytic tubules, clearly defining where the branch point of this structure will take place. So we found a mechanism by which bones are shaped, at least in the context of bone splitting, that it's interesting for fundamental but also more applicable contexts. And in fact, we tested a battery of drugs to identify compounds that are actually able to induce the formation of bone or to actually stimulate bone resorption. And as you can imagine, this is highly important in the context of osteoporosis, for example. And by just simply looking at this splitting point, we can actually screen for compounds which have the ability to form or to resorb bone. So I talked now about thin as the next leverage or the model of excellence in regenerative biology, but I'll also talk to you a little bit about the heart. And this is highly important because with aging populations, the prevalence of cardiovascular diseases, in particular ischemic heart disease, become extremely important. And what we do here with Zebrafish, so the experimental model that we use is cryo-injury And this is basically using liquid nitrogen cooled probe. And we expose this heart and we touch the tip of the ventricle. So basically we're inducing cell necrosis there. And then we can track the regenerative process. So in very simple terms, the heart is composed in three main cell layers. So we have the endocardium on the inside, we have the epicardium on the outside, and in the middle we have the myocardium. So this is the beating part. So it's the muscle part of the heart. So it provoke a necrosis, and very fast the inner and the outer layers, so the epicardium and the endocardium, they activate and they migrate. So they form a regenerative scaffold. And very fast, also, there is still the formation of a scar in the middle, in the injury area. So we have a scar, we have regenerated inner and outer layers, so now we need to form new muscle. And this happens gradually over time as this scar becomes resolved. So the cells, the cardiomyocytes, which are the beating muscle cells, they re-enter cell cycle and they slowly regenerate and restore the cardiac tissue. Many things are happening during this process. We have fibroblasts taking place, deposition of ECM. So many people are focused on different aspects. And one particular aspect that is very important, and people were talking about the immune response, is this compartment there. So I became a bit interested in that. So we knew the immune response was important because if we block it overall, we do not have regeneration. But if we exacerbate, for example, inflammation, there's also not regeneration taking place. So we asked which immune aspects actually modulate regeneration. And just to give you a notion, this is fairly doable in zebrafish because there are many, many tools available to study the immune response and the different compartments of the immune response, such as inflammatory cells like neutrophils, also macrophages. So we have inflammatory and anti-inflammatory, also T cells and eosinophils, and most of the cells that you can think in mammals, you will find them in zebrafish. And most likely there will be tools to look at them, to trace them, to manipulate them so we can actually ablate them and see what happens, and also to manipulate gene expression in a cell-specific manner. So I just put this slide here so everybody's on the same page. We have different compartments of the immune response. On one side, we have the myeloid cells, such as the granulocytes like neutrophils, also the macrophages, the dendritic cells, and these compose innate immunity. But on the other side, we also have the adaptive immune system, which is mainly composed of T and B cells. And we have them in zebrafish in a highly conserved manner to those of mammals. And just to understand, we also have antigen presenting cells, which are typically macrophages, dendritic cells, and B cells. So what are antigen presenting cells? So these are the cells that are actually responsible to activate T cells. So macrophages, dendritic cells will, together with antigens, will activate specific T cells. So we decided to look at this in the heart, what was happening. So we looked at different antigen presentation genes. So we're trying to understand which cells are being antigen presenting and activating T cells in this context. So we found out that indeed macrophages were becoming antigen presenting. So you can see here, this is a macrophage marker, and you see that it's expressing the antigen presentation markers. But interestingly, we found that not only immune cells were expressing these markers, also endocardial cells. So these are the inner, the cells composing the inner layer of the heart. So this was a novelty. And we saw that also by sorting these cells with different reporters and performing gene expression analysis. So they were indeed activating the expression of these genes. We looked at what was happening in terms of T cells, and they highly accumulated in the injury. So you can see here, peaking at day 7, and they were always in contact with the endocardial cells. So this was a strong indication that there was a tight crosstalk between these two cell types, and that endocardial cells were sustaining the activation of T cells. So what we did back then was to target antigen presentation. So in hopes to, if we block antigen presentation there, we won't have, or we have an effect on what happens in terms of the downstream T-cell responses. So we generated mutants for particular molecules, so CD74. I'm not going to go into details, but the idea is that by CRISPR-Cas9, we blocked antigen presentation and the downstream responses, of course. So we found that in this loss of function model, we had a problem with T cells infiltrating the injured tissue. So this comes in hand with the idea that antigen presenting endocardial cells were sustaining the activation of these T cells. And this again comes in line with a problem with the endocardial cells themselves to penetrate the injured tissue and to proliferate. So there's an important role of antigen presentation here in activating T cells, and probably T cells are also required for antigen presenting cells, or in this case, endocardial cells, to proliferate and to regenerate the tissue. So the question is, okay, to have regeneration, we need to have the formation of new tissue. And in the heart, the most important tissue, of course, is the beating tissue, which is the myocardium, which is composed by cardiomyocytes. So we looked at cardiomyocyte dedifferentiation. And as I told you in the beginning, for regeneration to take place, most of the times these cells need to dedifferentiate to become more immature in order to be able to proliferate and form new tissue. So we looked at, in this antigen presentation, loss of function model, and cardiomyocytes had a problem with dedifferentiation, and this came accompanied by a problem in cardiomyocyte proliferation. So in fact, these cells were not being able to proliferate and to form new cardiac muscle. So, but back then and nowadays, actually, people say, okay, but T cells are also bad when you have an injury. So, and here I'm showing that they're most likely good. So the ongoing hypothesis is that there is a high T cell heterogeneity, and the balance between the different types of responses, so good T cells and bad T cells, this is what actually defines what happens post-organ injury. Do we have regeneration? Do we have even further tissue damage or not? And the thing is that there is a likely shift towards tissue damage as opposed to organ regeneration taking place with aging. And this comes, of course, with the crosstalk with the inflammatory potential, as I mentioned before, and the ability to resolve this inflammation. So I'm particularly interested in aging and what happens in aging, and there are multiple models established in the field, zebrafish models to study aging. We can look at CLO2, and this is an anti-aging hormone, actually not model here, that declines with aging in humans. So it's actually a nice physiological aging model. We have other models, so we have TERT models, and this is more like what I call a cytogenetic model because it relates to chromosomes themselves in the telomeres and the telomerase. That is actually important to maintain telomeres integer. And we have more newer models such as CELEST-R1A, which was established as a stem cell-related aging model. And an interesting thing, and just picking up on something that Mohammed said before, is that you can also use zebrafish to study what cells are doing, because there are multiple established models, immunosuppressed models, that you can use to actually perform transplantation studies. And this is what Mohammed was talking before about transplanting cancer cells that you can actually use to try to do some personalized medicine by testing different drugs and see what works more on different patients. And I'm also interested in using it to transplant different T cells and see what they are doing. And don't worry if you don't understand this, maybe you'll understand after the following talks. So this is what I'm interested in now. And to address some questions, I have some tools ongoing, like different loss and gain of function tools to study in a cell type specific manner the role of different T cell signaling. I also am particularly interested in which antigens, so these are the upstream molecules that are responsible to activate different T cells. So I'm interested in identifying which are the good ones and the bad ones. So I have cell type specific knockouts to block antigen presentation in particular cells, and also to pull down these complexes and try to identify which are the different antigens. So I saved a few last slides to show you more advantages of Zebrafish in regeneration, but this is applicable to anything you can think of. So one advantage is also, of course, the fact that you can use Zebrafish to perform live imaging, and this is very immediate when you think about larvae or smaller animals. But if you think about adults, and the fin, for example, is very suitable for this. And just some videos showing you that you can actually track cell migration, for example, here as well. And you can easily perform optogenetics. So there are multiple lines and people can generate newer lines to photoconvert, for example. So this is a line where you can, if you can photoconvert a particular molecule from green to becoming red, and then you can track what's happening to the new cells that you photoconverted, for example, and this is easily done in the fin because it's external. The scale is another interesting model to study bone regeneration. And it's very interesting because it's also very superficial, so you can actually track it and perform time lapses on it. So this is a time lapse of regenerating scale. And you can see here, in this case, it's just an example how it goes with ERK activity, for example. And a recent thing that was described was this tessellated lymphoid network. So this was 1 year ago, it was published. And this is a superficial network below the scales also, which is actually considered now as the major reservoir of T cells, similar to what happens with lymph nodes in mammals. And this gives also a very nice, interesting system for live imaging, because these cells actually travel in this system, in this network. And you see here, they're very fast. So these are minutes. And here in a zoom. So these are T cells in green and CD4 positive T cells in red. And in the heart, for example, this is an additional a challenge, because this now we come in the area of internal organs. This is a challenge for live imaging. People started doing explants on sections. So it's live sections of the heart. And here I hope you can appreciate a cardiomyocyte dividing. So this is important for heart regeneration. And in the context of immune cells, I developed a system where you can use the entire heart to image their behavior. And this is just a proof of concept. So these are neutrophils, and you see that in a normal situation, in an injured heart, they are having a particular behavior, so they're moving. And if you stimulate these hearts with a pro-inflammatory drug, you see that these cells become kind of crazy. So it's also a platform to try to identify compounds with the ability to immunomodulate cells and control inflammation. And you can actually analyze different behaviors, including phagocytosis, as you can see here. So these are macrophages phagocyting in the regenerating heart. So with this, I hope I gave you a nice overview of what you can do with Zebrafish in terms of regenerative medicine. Of course, there's much more to do, and there's much more in the field, which in 30 minutes is difficult to put everything. And with this, I want to thank, so these are the people that were involved in the parts of my own work here. And I want to thank you all, and I take questions.
João, thank you very much. It was fabulous. It was really fabulous seeing all the possibility we have to use zebrafish, and yeah, it was fabulous. And especially, I know that people, including myself, love seeing cell in a real manner and in real life, how they deal, how they move, how they
Me too, me too.
how they act. Actually, maybe before giving the floor to the participants, I have some question and curiosity. I will start myself, okay? I have some question and curiosity.
Sure.
Uh, 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 so myself, I work in the role of genomic instability and chromosomal instability in helping cell to resist to chemotherapy, but also to move. So what I showed in other models that cells with extra chromosoma move faster. And when you talk about the generation, meaning that the cell has to undergo division, rapid division. So did you notice any problem in cell cycling of these rapid or rapidly dividing cells? So did they follow an exact like or a nice division, or they face some chromosomal instability, as I was saying? And also, I mean, just by checking the DAPI, I mean, if the nucleus were the same, if they were positioned in the in a way same places. Also, I have another curiosity. So when you talked about regeneration in tail and in heart, so in a really basic biophysical way. So regarding tails, you have like a loss of something. You see what I mean? So you lost something. So the cell undergo a path of several pathways to recover. But in the heart, when you put your like a cryo uh, not injection but cryo
Cryo injury. No,
yes.
you perform cryo injury, do cryo probe. Yeah.
So you still have something. You see what I mean? So you still have something in the heart. It's dead cells or dead tissue, but you still have something. So I mean, is it the same pathway engaged here? I mean, just comparing something that are definitely lost and something that is dead but still here. You see what I mean? So
Yeah.
are they the same? I mean, pathway?
So
Yes, please. Yes.
so let me try to answer the first one first. So I understand. And so to give you an answer, I don't know. I never looked, and I'm not sure somebody has. Probably they did. Uh, I'm not really in the chromatin field, but it makes sense what you're saying, of course. And people, there's also this regeneration versus cancer debate, right? But to give you a straightforward answer, I don't know.
Nice.
For the second, yeah, you're completely right. In fact, there's some groups in that actually do not perform cryo-injury. They perform cardiac resection.
Okay.
In my view, I prefer the cryo-injury model because in more it reflects more what happens in a clinical setting, right? So if you have a myocardial infarction, you're not losing part of your heart. You are in fact having dead tissue there. And this creates an additional problem for the organism, which is, okay, we have to form new tissue, but we have to get rid of the bad one, let's say, the dead one.
Good.
So in fact, this comes with different pathways being activated, of course. And especially if you compare the fin and the heart, the cells are not the same, right? And you have, for example, different patterning mechanisms. You have in the heart, you have scar, transient scar. In the fin, you don't. So you have many differences, but you also have general responses. So in terms, for example, of the immune response, at least the initial response is very similar, and the triggers are very similar. what happens later on, it's what starts to differ. Yeah, in terms, of cell cycle reentry, for example, if you compare the heart and the fin, there are different mitogens taking place. So you have, for example, neuregulin triggering cardiomyocyte regeneration, and you have other mitogens in the fin. But that's why I think these are very complementary models, because you can identify organ-specific responses and regenerative responses, but also global ones. And knowing the similarities and the differences, I think it's very relevant.
Yeah. I have, I think, a question here. Thank you very much, João. It was a really fantastic presentation, and
Thank you.
I look forward to meeting you in person, like in the.
Yeah. Me too. Next year.
So, yes, Zebrafish model. So we will talk about the model like later. So sorry. Is there application in nanomedicine? Yeah. So I do assume that Vincenzo will give us.
Yeah.
Yes.
I don't work with this, but I think I came up across with a few papers on it. But I cannot give you a full. So this is completely out of my topic. But yeah, but for sure, if we Google Zebrafish and nanomedicine, that there will be something coming up.
Definitely. I have another curiosity, maybe before I'll still go giving the photo to people. I saw, I don't remember really in which way, but still regarding the cell cycle, I saw, for example, when we injure the skin of the zebrafish, so the cells go rapid division, just for the cicatrization.
And sorry, we're talking about injury on the skin?
On the skin, yes. So
yeah.
I don't really remember where I saw it, but what I kept is that the cell undergo rapid division for the cicatrization, and this rapid division so gave the as I said before, like a very unhealthy cell. So, but the goal was just to recover the injury, and then the cell undergo death, and they was little by little replace it by a more healthier cell. So yeah, it would be fantastic if, in a way, we could see this in your regenerative model of heart, or I don't know.
No, we also have something similar, because apart from the necrosis, and I didn't tell you, there's also apoptosis. So, and these cells are taking part in the regenerative process, but then at some point they undergo cell death. But yeah, it's a bit complex.
Yes, very nice discussion, very nice presentation as well. Thank you very much. Any question from the audience regarding the work of João? You can open your mic, or you can raise your hand, or whatever.
Mohammed.
Yes, please.
The question is
I'm not hearing you, Marco.
And how could we understand the loss of regeneration in the hyperglycemic model? It's possible to study this model.
So you're asking me what happens in an hyperglycemic model, or
an hyperglycemic model is you can't understand the loss of the regeneration process.
Yeah, I think for sure, because not only hyperglycemia, but metabolism in general. So there is, and there are many people working on this actually. There is a strong involvement of metabolism in regeneration, as you can imagine. And so these regenerating cells, for example, in the heart, they need to undergo they need to shift their metabolism to glycolytic metabolism, for example, in order to regenerate. Otherwise, regeneration doesn't happen. So, yeah, I don't know if this really answers your question, but
yeah.
Okay.
Mohammed, please,
yes,
Mohammed,
please.
I have a suggestion. Thank you for your presentation. It's very important. In fact, it isn't a question, but it can be a suggestion for you, Professor.
Yes.
As I work on the nanoparticles, and they have effect as regenerative agents, so I suppose or I demand, have you tested any simultaneous effect of such agent? I mean, have you tested the effect of nanoparticles, for example, on zebrafish model, since they can help, they can improve, they can ameliorate the generative action of cellular model or zebrafish model.
Thank you. No, I haven't done it. So it's interesting, and it's something that we can do for sure. At the moment, I'm more interested in the T cells themselves, and I didn't explain, but so the idea is that T cells are a key component of the immunity, and actually in the organism itself, that we can actually harness because it has a memory response as well. So I'm more trying at this point to try to predispose the organism to be more pro-regenerative. But yeah, of course, Zebrafish offers a very good system and platform to test everything, including the nanoparticles, for sure. This is very interesting.
Okay, João, thank you very much. We have to move.
Thank you. Yeah, of course.